Modern definition of viruses. By the 1930s, viruses were classified according to the host that they infected. Three groups of viruses, plant, animal, and bacterial, the bacteriophages, were recognized. They were identified and stored in liquid filtrates derived from the leaves, lesions, or juices of their hosts. As methods became available to purify viruses from the complex mixtures of chemicals found in these fluids, the chemical composition of a virus could be determined and used to classify it. When, in 1935, Wendell Stanley succeeded in obtaining a crystalline form of the tobacco mosaic virus, this form was seen by some scientists as filling the gap between the living world of reproducing viruses and the chemical or non-living world of molecules and crystals. In truth, the ability to form a crystal simply reflects the structural uniformity or symmetry of all the viruses in a population, together with a surface complementarity that permits an orderly aggregation of virus particles. German technology in the 1930s provided a new tool, the electron microscope. Instead of visible light, it employs beams of electrons focused by magnets, permitting the useful resolving power to go from 1 to 2,000 diameters magnification to 300,000 diameters. Now the submicroscopic world of viruses could be visualized, and electron micrographs showed the beautiful symmetry of viruses predicted by the crystallization of the tobacco mosaic virus. Today, chemical composition, symmetry, and structure make up the basis of virus classification. Almost all viruses are composed of two parts: a nucleic acid core containing the genetic information and, surrounding and protecting this core, a coat built from repeating protein subunits and, in some cases, further encased in a lipid or a fatty envelope. The virus's genetic information may be encoded in deoxyribonucleic acid, DNA, as it is for all non-viral living organisms, or in some viruses it may be encoded in ribonucleic acid, or RNA. Both DNA and RNA are long, linear polymers composed of subunits, monomers, that are linked together by strong chemical bonds. The monomers, called nucleotides, are each composed of a base, a sugar, and a phosphate group. The type of sugar gives the nucleic acid its name. In DNA, the sugar is deoxyribose, and in RNA, it is ribose. DNA is usually found not as a single polymeric strand, but as a double strand with two polymers wound about each other in a helix. The sequence of nucleotides in a DNA or RNA polymer determines the genetic information, and the chemical composition of this genetic information, whether it is encoded in DNA or RNA, is used to classify viruses. Viruses may contain double-stranded DNA, herpes viruses, single-stranded DNA, parvoviruses, double-stranded RNA, reoviruses, or single-stranded RNA, poliovirus. The RNA viruses are unique in that they are the only living organisms that use RNA to store their genetic information. All other reproducing forms of life employ DNA. And that's the modern definition of viruses. Have a great day.